so it's going to be 0. Now in milligrams, sorry, we were doing it in milligrams per litre. We have to convert that to milligrams per litre is the same as micrograms per mil, I think. So 0.8, 2, 3.2, and 4. Okay. I think that's right, guys. Just milligrams per litre. Just check your conversion of units, okay? Milligrams per litre. Let me check that. Okay. So anyway, you have this is your concentration. Okay. So these are the numbers you're going to have on the x-axis. Okay. But you need to determine the average extinction values here. So this is really easy, okay? So in Excel, you have your values here. You're going to hit the function button. So the function button is beside the toolbar here, okay? And a little box will pop up, and the first one in there is the average. So you're going to click OK and highlight the 2 cells that you want to determine the average of, all right? And I'm going to copy that and copy it all the way down, even for the unknown, because we need the unknown. We need the absorbance of the unknown to determine the concentration of the unknown. Okay, is everybody following that so far? Can you answer me, please, if you're still there, guys? Am I talking to myself? Is anybody there?
Yeah, sorry. Okay. Some people are answering in the chat.
Okay. All right, so that's our first part. Okay, now remember, this data is not the same as the data you were provided with. So you're going to have to do this using the same logic for your own data. Okay, so I'm going to copy these 2 columns. Okay, copy these down.
Sorry, Rachel, is this example available anywhere? Because I can't see it on either the biology lab page or the biology page that we have.
No, it's part of your practical, and in normal years you would have to do this on your own. There will be tutorials. Okay. This is what the tutorial is for, to go through this. Okay, so you can take pictures of this, guys, as we're going along, or you can take it down. But like it's up to yourself to kind of, record anything that you need to know for doing your own report. Okay, so this isn't a kind of an incentive to attend tutorials and things when they're on, because we do things like this at them. Okay. So now I have my bromophenol blue concentration. Okay, so that's in one column, and I have my extinction values on the right hand column. Okay, I'm going to highlight the 2 sets, not including the unknown, okay, because we don't need that on the graph. We're going to use the equation of the graph to determine the concentration of the unknown solution. Okay, so you can see I'm highlighting the titles, okay, the title of the column and the numbers. So on the left I have my bromophenol blue concentration corresponding to tubes A, B, C, D, and E, and then I have the extinction values. So I'm going to highlight that. I'm going up to insert. Okay, I'm going to insert an XY scatter, okay, over in the charts. So insert XY scatter, and there's my graph. Okay, now I need to dicky that up a bit. Okay, it's not suitable the way it is. So I have to put labels on my axes, etc. So just to kind of look at this for a second, make sure your extinction values are on the y-axis. Okay, make sure the extinction is on the y-axis. Now, if you see up here, there's a button for quick layout. If you click on that, it'll give you options to give your axes titles. Okay, so I'm going to go for this one. We don't need a title on the top, okay, because when you do your report in Word, you're going to write the title underneath it. You're going to type a proper figure caption, whatever wavelength is determined, okay. And this is not sorry guys, I don't want all those data points. Delete those. You just want literally your point. Okay, so you can see the point. You can see that there's nice and linear. Okay, and you can get rid of this as well. Now, if my thing would let me go down a bit, yeah, the X title. So the X-axis title is going to be bromophenol. Concentration, and that's going to be in micrograms per milliliter. Okay. Okay. Is everybody okay so far? We're all following how we did that in Excel, how to plot the graph. Type in the top box or put on your microphone, guys. You need to know that you're staying with me for this. Yeah, okay, great. Okay. Anybody else having any problems or anything what I've done so far? Is it clear? Okay, so for anyone who missed how to insert the graph, okay, I'm highlighting my data, not including the unknown, okay, because we can't put that on the graph because we don't know the concentration, okay. So I'm highlighting the data and then going up to insert, and I'm going over to the recommended charts, and I'm going to choose a scatter plot, okay. So click on that and I'm going to pick the one with the points on it, okay. Now I have to tidy up the graph, okay. See the quick layout? There's options here for putting in titles and things. So I don't want the data points on the graph. Take them off. And I don't want that there. And I don't want that there. I just want to label the axes titles. You can tidy up the axes as well if you want to, like, get rid of the grid lines, turn them inside and stuff. It'll make your graph look neater. Okay. So a little box will appear over here. See here, tick marks. You can put them on the inside. Okay, and the same for the other one. Tick marks inside. Okay, no, sorry, I meant inside. Okay, so that's how the graph goes in. Now, the next thing you need to do, I'm going to delete that. Okay, the next thing you need to do is you pick one data point on the graph. You're going to right click on that. So you see all the data points will be highlighted. Okay, I want to add a trend line. So that's how we put in our best fit line. You add trend line. So a linear trend line, you can see it's already selected, okay, and I want to set the intercept to 0 so that it goes through the origin. I want to display the equation on the chart, and I want to display the r squared value on the chart. So I want to do all of those things, okay? So you see that popped up just above the trend line. Y equals 0.1503x. That's the equation of the line, and the r squared, that just shows you how linear it is. So it's 0.995. So that's very good, okay? Now I just want to format the actual line. So I want to make it a solid line, and I want to make it black. Okay, solid. Okay, so now I have a solid line, okay? Is that clear to everybody? So once you have your equation, you can determine the concentration of your unknown. So in this case, our unknown, we're trying to determine the concentration of it, but the Y value is the extinction. Okay, so if I put the Y value that I know is here, so just to hang on, this is my equation of the line 0.1503x. Okay, I know my Y value, so my 0.31 equals 0.1503 times x. Okay, everyone following this?
Yeah,
so
thanks.
You can see, when you have an equal sign, if something crosses the equal sign, it's going to change the meaning of it. Okay, so if on the right hand side of this equals, it's a number multiplied by x. If we bring the number to the left of the equals, it will become a divider rather than a multiplier. Okay, so we have 0.31 divided by 0.1503. That gives us our x value. Okay, so just do that there if you have a calculator or your phone or whatever. So what answer did you get for the bromophenol blue concentration? Yeah, perfect, guys. That's how you do it. So 2.06, okay? So if you kind of round that down to just 2, okay? If you go to, we're lucky that we have a data point that's 2, okay? So we know that the extinction value for that bromophenol blue concentration is the same, okay? So that kind of suggests that your data is good. So if we go to the extinction values, just look at the y-axis for a minute. The extinction value is 0.31. If we go up to 0.31 on the graph and imagine that we're going across to the line, okay, you can see there's a data point roughly at the same point that hits the line, the best fit line. And then when we go down to the corresponding value on the x-axis, it's 2, okay? So you should be able to check your equation corresponds to the data that's presented on your graph. Does that make sense to everybody? You can do a quick look-see. So for instance, if the extinction value, just say for instance it was 0.4, okay? Looking at the graph, what do you think that would correspond to in micrograms per mil of bromophenol blue? If it was 4, what do you think the answer would be? Sorry, the extinction value was 0.4. So go up your y-axis to 0.4, go across to the trend line, okay, and go down to the x-axis. What number do you think would be for the bromophenol blue?
.6 maybe?
Yeah, in around that. Okay, so use your graph as a guide, okay, but the actual equation will give you an exact number, an exact numerical value, okay? So is that clear to everybody? Do you know how to plot your graph for your report?
Thanks, that was very helpful.
Okay, just somebody's typing a message there, but it's taking a long time. Is everybody okay with that? I know there's only 6 of us here this morning, but if you want to ask anything, guys, now's your opportunity. So, okay. Okay, so when you're done, when you have your graph plotted and tidied up and all the rest of it, you just copy that into Microsoft Word. Okay, and that's what you're going to discuss in your lab report. So everything you need is here. Okay, so you need to do this in Excel. Okay, you'll have this set up here. Your tube, what name is on the tube, the amount of water in the tube, the amount of your bromophenol blue, and the concentration of that. So you'll use these to determine the dilution factor essentially, and that will be used to determine the bromophenol blue concentration of your unknowns. So you can see that's 0 for A, B, C, D, E, and F because you only have one unknown solution. Okay. So essentially, when you determine what concentration is in each of the tubes, you'll fill that in. So bromophenol blue concentration, there's a column there for that. And then the extinction values, you'll have your sample data set, you get the average of that, and then you plot your two columns against each other. So the bromophenol blue concentration and the average extinction value. So just copy those two columns down further in your Excel worksheet. Okay, and plot the graph, make sure the extinction values are on the y-axis and the bromophenol blue is on the x-axis. Plot your data points, add your trend line, make sure it's a linear line, make sure it goes through the origin, so you set the intercept to 0, 0. Show the equation on the line, show the r-squared value on the line. Okay? Okay, guys, so I'm going to leave that now. Okay, is there anything else before we leave it? Anyone want to ask anything? Yeah, that's for the curve where you're comparing the two dyes. So the bromophenol blue and the methyl orange. So if you look at this is the data that's referring to. Okay, so there'll be an absorption maximum. You'll get a kind of a bell-shaped curve. So just wait, you'll get something like this. Okay. So you need to say why the values are different. That's because they're two completely different colours. Okay, and there will be an explanation for that in your manual. No, if it's not on the y-axis, Sean, but the best way to do that is, see the way I have, I've put the bromophenol blue on the left and the average extinction values on the right. Okay, so when I plot the graph, that's the way it goes. If it doesn't turn out like that, switch the columns, switch the order of the columns. Okay, or if you see that I can, the plot area. No. Yeah, no, you'll have to, if it doesn't, like, there's only one of two that it can be. So when you have your data arranged in Excel like that, you only have two columns of data. Okay, so the bromophenol blue on the left column and the extinction value on the right. And if it doesn't work out like that, change the order of them. But you need to just check on that yourself. Okay. Any other questions before we leave this? Okay, guys. So, if we're done with that, we'll do a quiz. Okay, so if we want to use Mentimeter, these are just some questions on your lecture on antibiotics. Okay, so if you go to menti.com and put in this code 4308580, you all see that? So make your selection there. Okay, guys, before anybody else want to make a selection? Anybody else? Okay, well, the 4 of you who selected cell wall, that's incorrect. Okay, polymyxins work on the phospholipid membranes. Okay, so you're all wrong on that one, guys. So go back and have a look at your notes. Okay, it's moving on to the next question. Okay, so do we only have 4 people participating in the quiz? 5. Okay. Okay, guys, so sterilization is incorrect. Okay, selective toxicity is the right answer. So it's basically like the magic bullet. We want to kill the disease-causing microbes, but we don't want to harm the host tissues. Okay, so well done, everyone who got that right. And the last one now of this set. Which of the following ailments would antibiotics not help to heal? Anyone else? Okay, so the correct answer is flu because it's viral. Okay, bacterial meningitis, antibiotics will kill bacteria, and botulism is caused by Clostridium botulinum, which is a bacterial infection as well. Okay, so the only one that antibiotics wouldn't help is the flu. Okay, see, anyone who selected meningitis or botulism, you need to go and read your notes. Okay, so we move on to the next one. So you need to put in a new code now, guys. So go to menti.com and the code is coming up now, 6483153. So the genes encoding antibiotic resistance are found where? Anybody else, guys? Can you speed up a bit with your answers? Okay, so only one person got this right. Correct answer is plasmids. Plasmids is the right answer, okay? It's not chromosomes, okay? I know there's other genes found on chromosomes, but not the ones encoding antibiotic resistance, okay? And ribosomes is incorrect completely, okay? So, well done to whoever you are who got plasmids. That's the right answer, okay? Next question. Which of the following features can pump antibiotics out of a bacterial cell? 4 options. Anybody else? Okay, so efflux pumps is the correct answer. Okay, fimbriae are just like tails that the bacteria swim using. Plasmids are inside the cell. Okay, they don't really do that type of work where they pump things out of the cell. Membrane pore, not necessarily. It's just a kind of opening in the cell wall. Okay, so efflux pumps is the correct answer. Okay, so well done to the 4 of you who got that right. And last one in this set. So, what is the mechanism of action of penicillin? So, how does it actually kill microbial cells? See 4 options here. Okay, well done. So, inhibition of peptide-glycan synthesis is the correct answer. So, well done, everybody who chose that one. Okay, inhibition of protein synthesis is incorrect. Okay, so we'll move on now, guys. We have 3 more questions. So, you need to enter a new code. Okay. So the new code is 6169715. This is a nice and easy one. The building blocks of polysaccharides are which of the following? 4 options. Okay, so whoever selected amino acids, that's incorrect. Amino acids are the building blocks of proteins, okay? Nucleotides are the building blocks of your nucleic acids, okay, and fat and the bases. Fatty acids are the building blocks of lipids, etc. So sugars is the correct answer, okay. Moving on to the next question. So, which of the following is a protein found in ligaments and tendons in the human body? Okay, 4 options. Which of the following is a protein found in ligaments and tendons in the human body? Okay, well done, everybody. It's the right answer. Collagen is correct. Okay, so last question for today. Which of the following enzymes degrades starch? Anyone else? Okay, so protease is incorrect. Whoever selected that. Okay, protease degrade proteins, lipase degrade lipids. Okay, amylase is the correct answer. It's the one that degrades starch. Okay, and elastase is a different enzyme. It's not associated with any of the big molecules. Okay, so well done, guys. That's it for today. Is there anything you want to ask or anything before we finish up? It's your last chance now. Okay, guys. Good luck